All right. Good afternoon. So I'm going to be a little silly and I'm going to try to create some engagement. I'm going to say good afternoon. I'd like the audience here to reply back so that the online audience could reply back virtually. So I'm going to say good afternoon. One more time, harder. Good afternoon.
Yeah.
Good afternoon. All right. Thank y'all. So I'm here to present Francis Boquet. He is a medical research specialist in the core lab in the Division of Immunology at Tulane National Biomedical Research Center. He holds a Bachelor of Science degree from Nicholls State University and studies congenital cytomegalovirus infection and placental transmission in a non-human primate model. And I'll also add that Francis has a very unique skill set of being innately curious about the work he does. And he tries to really do right by the precious animal samples that he works on. So you'll get to see some of that work here. And outside of the lab, he enjoys roasting his own coffee to fuel late nights in the flow cytometry core, which he has statistics on having a lot of that. So give it up for Francis.
Thank you, Matilda, for that great introduction. And I'm very happy to be here today and being able to talk to you about how collagenase type can influence both immune cell recovery, viability, and surface phenotype of leukocytes isolated from non-human primate lung tissue. So for the initial aim of our project, we wanted to improve our viability and cell recovery in this tissue, which for our lab has been a main pain point, so much so that there have been multiple lab meetings where we discussed, leaving it out of our panels and not sampling it anymore. But it's always been important to me to keep it in our panels and to keep studying it and to try and do better. So having said that, we decided that manual dissociation methods weren't working for us, so we wanted to enzymatically digest this tissue and see if we could increase our viability and decrease this kind of range that we're having with it. And doing that, we did see that we did get more consistent cell recovery across our time points. However, this added a whole nother layer to things. It brought up the question, since we're now adding an enzymatic digestion to our protocol, how is this going to affect our downstream data, more specifically our flow data? So we kind of had some options when we were selecting for collagenase type, and we landed on these two, collagenase type 4 and collagenase type D. We chose type 4 because it's what we normally use in our other enzymatic digestions like placenta and decidua. And I really wanted to try the type D because it has this, has this reported lower tryptic effect on the tissues. So really they're derived from the same biological source, Clostridium histolyticum. And their common uses are just a wide range of tissue types. Their main difference is in how they're prepared. Collagenase type 4 is a more crude preparation, while the type D is a more filtered preparation, which is reportedly what gives it its lower tryptic activity. And I just had some other activities that they have. But to move on, to kind of give you an experimental overview of what we did is we took a paired lung sample. So we took the left lung and the right lung, and we perfused each lung with either collagenase type 4 or collagenase type D. And from there, they were processed in parallel. So we used a Genomax octodissociator to help liberate leukocytes from the tissues. That was followed by a Percol gradient to enrich for lymphocytes. We stained a 15-color panel for viability, lineage, activation, and chemokine markers. The samples were acquired using a BD-LSR Fortessa X20 and analyzed using FlowJo. So to just kind of give you an overview of the panel that we ran, I broke it up into 4 categories. We ran markers for lineage, which included CD45 as like our pan-leukocyte marker, as well as T cells, monocytes, and macrophages. We then have our differentiation and activation markers. We had some chemokine markers, and we have a marker for viability. So to just kind of give you an overview of the conditions we ran an unstained, we ran a condition for each collagenase type, and we ran 2 FMOs, one for CXCR3 and one for CCR4. So in evaluating this data, the first outcome that we looked at was our total cell recovery and our viability. And as you can see here, our viabilities were pretty comparable, both being above 80%, The real difference we saw was our cell recovery in the collagenase type D was two times more that of the collagenase type 4. So this led us to our next question. We've acquired more cells using this collagenase, but are they our CD45 positive leukocytes that we're after? And what's the quality of these cells? So to determine that, we looked at our flow data. And the first plots we looked at were our CD45 frequency. And in this, we saw that our type D was able to increase our CD45 positive live by over 25%, while also reducing the proportion of dead cells and CD45 negative live events. However, when we look at our median fluorescent intensity, we can see that the type D condition has a decreased fluorescent signal despite having an increase in frequency. Now, despite this lower signal, we do still have good enough resolution to accurately place our gate around our CD45 positive live cells. The next thing we looked at was within our CD45 positive live population. We wanted to see how does collagenase type D affect our CD3 positive cells. And what we saw was that we had a 35% increase regarding our frequency in the collagenase type D condition. However, we saw the same trend in MFI where although we have this increase in frequency, we see this decrease in MFI. Now, I will say that the resolution is getting a little close here in our type D condition. We can still separate the negative from the positive, but I really wouldn't want to see it go down any further than that. So within our CD3 positive population, we decided to look at our CD4 positive and CD8 positive T cells. We saw that they were pretty similar, slight differences here and there, but the real difference came when we looked at our MFI. So our CD4 positive in MFI had a threefold increase compared to the type 4 condition. And to me, this is consistent with an improved preservation or accessibility of the CD4 epitope, possibly due to type D's reported lower tryptic activity. Next, we looked at a few non-T cell lineages. This one right here is going to be our CD3 negative, CD8 positive natural killer cells. And we saw an increase of about 48%, which is pretty large when you look at this at under 3% all the way up to 50%. Next, we looked at our CD206 positive tissue resident macrophages. And our collagenase type 4 condition resulted in approximately a 4% higher frequency compared to our type D. Now, you might be saying, oh, 4%, that's not very much. But whenever I look at this graph, I can see that there's a population right here. I can gate on this population and I can see it. And when I look at this type D condition, I just don't see that. So to kind of give you a summary of what we just went over, in our collagenase type 4 condition, we saw that our cell frequency was increased in our CD206 positive tissue resident macrophages, while also increasing our CD45 and CD3 MFI. As opposed to our collagenase type D condition, we saw an increase in cell frequency in our CD45 positive live condition, our CD3 positive T cells, and our CD3 negative, CD8 positive natural killer cells, as well as a higher CD4 MFI. So to kind of move on into our activation, differentiation, and chemokine markers, we first looked at our CD28 expression on our CD4 positive T cells, and then all the subsequent markers that we're going to look at are also going to be on CD4 positive T cells. So what we see here is that our collagenase type 4 condition has approximately a 30% higher frequency compared to our type D condition. Now, this is going to be important to kind of keep in the back of your mind because next we're going to look at our effector memory, our transitional effector memory, and our central memory. And what we're seeing here is that our central memory is staying relatively stable while we're having this 30% jump between our two effector memory and transitional effector memory populations. And we believe that this shift that we're seeing is due to that shift in our CD28 positive population that we just looked at. So next, we'll move on to our chemokine receptors. We observed improved preservation of CXCR3 positive events in our collagenase type D condition. So typically, so actually, I want to back up. We not only did we see an increase in frequency in our events, we also saw a twofold increase in the MFI that we saw. So CXCR3 for us often requires an FMO for accurate gate placement. Typically, it looks like this. We don't have good resolution between our negative and positive populations. It's kind of just a smear. So, looking at that, you probably really couldn't say definitively where you would want to put your gate. But looking at this, I think that this type D condition has increased our resolution to a point to where it's not to say that we wouldn't need this FMO anymore, but it would definitely reduce our reliance on it for accurate gate placement. So next we'll look at our CCR4 expression. And like our CXCR3 expression, we saw an increase in frequency and, or not in frequency, I'm sorry. We didn't see an increase in frequency, but we did see a twofold increase in MFI, which I didn't show here because I thought it was more important to show you the FMO, because even though we saw that same twofold increase in MFI like we did in the previous slide with the CXCR3, that didn't give us the appropriate resolution that we would need to accurately place a gate without this FMO being there. So in conclusion, we saw differential preservation of T cell phenotypes. Our collagenase type 4 preserved expression of our CD28 population and our transitional effector memory, and our collagenase D type preserved expression of our CXCR3 and CCR4 populations. So in conclusion, we observed multiple lineage, differentiation, and chemokine differences associated with collagenase type that affected both cell recovery and receptor expression. And if this data was to be interpreted using only a single collagenase condition, it could lead you to different biological conclusions based on, which one you had chosen. So I think the key takeaway here is that collagenase type is a critical pre-analytical variable that can bias immune composition and immunophenotypic interpretation in your own experiments and in the data that you evaluate. And I just want to add on to that, so much so that we've begun evaluating what effects do all of our digestion medias have on all the various projects that we're doing right now. So this study did have several limitations. We did have a small sample size. Our samples were from different lungs, but the same animal. So like I said, it's the left lung and the right lung, but this does introduce the possibility of heterogeneity between the two lungs. We also lacked an enzyme-free dissociation method to characterize the effects we were seeing with enzymatic digestion. So in our ongoing work, we plan on getting more lung samples, replicating this, and seeing if we get the same results. We plan on taking the lungs and sectioning them into equal portions and treating them with collagenase type 4, type D, and adding that manual dissociation only method to hopefully help us characterize the effects of enzymatic digestion. I would like to acknowledge everyone at the TNBRC, especially my lab, who are all very big supporters of me, and I honestly wouldn't be here without them today. The Flow Cytometry Core, the animals and the animal care staff, and the funding that makes all the work that we do possible. And if we have time for it, I'd be happy to take any questions.
This work is so critical.
Thank you.
My question is hopefully very simple. Are you comparing like CXCR3 within your master mix, or are you doing sequential staining? Have you looked at?
Right. So no, everything is in a cocktail. So we do stain with a cocktail.
But that's phenomenal. The recovery of CXCR3.
Thank you for the talk. I just have a quick question, and I know this is asking a lot, so I don't expect you to say you've done this or will do this, but do you have a ground truth that you can reference, say through microscopy, where hopefully there's no processing steps, it's just like a frozen or paraffin embedded section that you can say, this is how many T cells we should have, and here's what we get with the different techniques?
Right, that's a great question, and I've actually been struggling with that. Like, how do we quantify like what is an effect of the enzymatic digestion versus even manual dissociation? I'm sure is going to have some kind of phenotypic effect, and so that's a great question, and I don't have an answer for that, but I would, if anyone has an answer for how we can really, characterize these cells and really believe that the phenotypic expression that we're seeing is the true state of the cell, I'd love to hear it. But yeah, I don't have a great answer for that right now, but I'd love to do it, I think it's important.
My question is similar on that, and I guess now it's a comment. Without going into like defining specifically like how many molecules are on the outside of the cell, like have you tried looking at the dying populations? And then have you also tried absolute counts, like to compare between the tissues? It like, for example, you're seeing these frequency differences. Is it because more of those T cells and NK cells are making it through in the collagenase D, or is it just that you have a lot more of them, period? Or has something changed on receptor expression, for example, of the myeloid cells that are missing now?
Right. No, that's a good question. And it is something that we've talked about doing, is doing absolute counts on these cells to really see like what the populations are and really help define that. I think that moving forward, we probably should do that. It's just this was something that we tested,
Pilot, yeah.
and we really, I don't think we were expecting to see these results.
Yeah.
And so now that we've seen these results, now we have this obligation to pursue them further and see, is this really a change due to collagenase type, or is it just some other variable that we're not thinking of? Are these changes real or not? Yeah.
Thank you.
Hi, Francis. Good talk. I have a rabbit hole question, which you won't like.
Oh.
Is there any, what concentration do we use? Because you're testing two, right? Are they
Right.
at the same concentration? One's filtered, maybe more pure.
Right.
What are we basing? That is do we titrate our enzymes?
Yes, no, so that's a great question, and it's something that I've also been thinking about. So the enzymes were reconstituted at 1 milligram per mil, and so they had the same concentration, but that's not to say that they have the same digestive activity, right? So in further studies, we definitely are going to need to find a way, because digestive activity can vary between lot and lot. And when I was looking at the spec sheets for these enzymes, they actually measure activity in different units. And so it wasn't easy to compare them. There is a synthetic peptide that you can buy and run an assay on. And based on the cleavage of that peptide, you can determine the activity of the enzyme. But that would have to be something that we do on a lot by lot basis. Yeah.
So I did a fair amount of work for BD when we developed the enzyme cocktail that we sell for tumor digestion, tissue digestion. And one of the ways I got around this was I used PBMCs and just ran them through the entire digestive workflow with each of the enzymes and looked at MFI pre-digestion and post-digestion. And so it gives you a reference point to whether or not you're getting passive cleavage on some of these epitopes. And I found it super helpful.
Yep, I'm glad you bring that up, because we actually we use trypsin in our placenta digestion, and I've noticed that the placenta has been having less CD8 and CD4 positive cells. And so I wanted to see, is trypsin contributing to that? So we actually thawed spleen cells, exposed them to trypsin, and tried to see if we could see the loss that we were seeing. And it's pretty much the experimental design that you just talked about.
Yeah. Yeah, but it'll work for the same for the,
Right.
yeah.
Yeah, it's a good idea. Thank you. Thank you.